So the idea for this afternoon was to have the presentation of Sandrine and Paul, which are quite technical. Then to discuss on the presentation and if we can go further into the breakout work, the definition of what is a multi-generational, transgenerational effects, try to write down some definitions and also go through some questions which are related to technical aspects such as experimental design, biological models, things like that. And tomorrow we will have the presentation of Cisco Saloma on the role of epigenetics and genetics in evolutionary biology, which will enlarge the discussion. Because Cisco could not present this afternoon because she's also presenting another thing in Group A. So if it's okay for you, we can start and with your presentation. So.
I wanted to so thank you for the opportunity to present this work from IASN and collaborators. I will talk about the importance of multi-generational studies for ecological risk assessment of ionizing radiation. And I will focus on invertebrate case studies after chronic gamma exposure. So ecological risk assessment relies on the link between exposure and effects and requires the use of reference levels. Several values are recommended for these reference levels, from 400 microrep per hour for aquatic ecosystems to 10 microrep per hour for generic ecosystems. This latter has been determined using a species sensitivity distribution with available chronic data, but even if it's of main interest, it remains some knowledge gap that can be underlined, such as the number of data, the endpoint used that are only mortality, morbidity, and the huge number of unused molecular data and the shift also between lab and in situ values of species sensitivity distribution. All this data encourages us to challenge this value with experiments with more ecologically representative exposure. So why and how more relevant exposure condition? Because of persistence of radiocontaminants in environment, coupled with somehow a short lifespan of environmental species, because also of multiple stressors in environment, and because of heredity of effects. All of those reasons can make the response of population different after multi-generational exposure, difference in abundance of structure and diversity of fitness. Thus, more relevant exposure conditions require deciphering mechanism, long-term effect assessment, and in situ studies. Among long-term exposure for different, different design can be used to answer different questions, as we talked this morning and a few hours ago. Long-term multi-generational exposure can be used to assess toxicity mechanisms, change in population response, and to assess also adaptive response. It can be followed by common gardens experiment to assess adaptive costs. transgenerational exposure, on the contrary, with a parental exposure and recovery for progeny, are run to decipher hereditary mechanisms, and in situ studies are of main importance to evaluate the whole system in natura. Today, I will focus on these two main points to illustrate the importance of this exposure. And I will first start with the example of Daphnia magna, published a few years ago in Aquatic Toxicology. Daphnia magna is a planktonic crustacean which exhibits cyclical parthenogenesis. Continuous gamma external exposure has been applied at different dose rates during 3 generations, F0, F1, F2, while brood size decreased only at higher dose rate at the first generation. It started decreasing at 70 microgram per hour at the third generation, with an amplification of the effect and an additional brood release delay for the high dose rate. So in that case, multi-generational exposure shows us an increase of damage and of their severity. It's that conclusion. In the second example, that a second example that I wanted to present deals with C. elegans gamma exposure. C. elegans is a quite well-known soil invertebrate, mainly self-fertilizing, useful to decipher mechanisms. After parental chronic exposure of wild-type strain with gamma rays, we found in the lab that brood size was reduced about 36%, mainly explained by a reduction of sperm counts. If irradiation continues on second and third generation, this effect appears at a lower dose rate, but with a similar severity. But with another experiment from our lab recently published shows that the population adapts herself after Indeed, chronic exposure of 17th generation of C. elegans mixed strains and mixed stage to 2 dose rate of gamma rays shows that population adopt a strategy to counteract the loss of spermatocytes previously demonstrated in hermaphrodites. Indeed, it increased the number of male, as we see on the graphic, above all at the higher dose rate. In addition, cross-condition exposure showed that population seems to be adapted at the higher dose rate, whereas it seems only acclimated at the lower one, since it tends to come back at the basal level once in the control condition. Then to sum up all these results, I had only time to develop on 2 examples, but more ecologically relevant exposure enable understanding increased severity all around throughout all generation irradiated, enable also to decipher mechanisms of heredity, assess adaptive exposure, adaptive responses and consequences, benefits or costs, and also enabled the setup of integrative indices such as genetic diversity that I cannot develop here. All of these to refine reference levels but also to demonstrate that in the risk assessment, the history of those is also of main importance. And that's it. Thanks.
Thank you, Sandrine. Shall we go on with Paul and then we have a general discussion?
Yeah.
Okay.
So, hi, I'm Paul. I'm a PhD student in Naylis Lab, and I'm going to talk about my work on the effects of ionizing radiation on DNA methylation changes in Arabidopsis plants that were exposed over multiple generations. Yeah, as you guys know, life is constantly exposed to some form of ionizing radiation, and with human activities, this can be significantly increased. Yet, as we can see, life persists in even the most contaminated regions, and therefore we're looking for methods of their adaptation. Now, in our case, we'll be focusing on the epigenetics, of course, and in my case, on DNA methylation. Now, short overview, yeah, maybe not necessary for you people, but DNA methylation can have an effect on the expression, as you know, depending on its location in the gene, being it in the promoter region, where hypermethylation generally leads to a reduced expression, and hypomethylation to the reverse. Gene body, however, is not as well studied yet. Additionally, DNA methylation can have an effect on transposable elements, which are sequences that are able to jump about in the genome, thereby creating changes in the genome and higher genome instability. Hypermethylation of these regions leads to lower expression of these transposable elements, and therefore, hypothetically, an increased genomic stability. Now, so what we did is to study the effects of ionizing radiation on DNA methylation. We exposed 3 different generations: the parent generation, generation 1, and generation 2, to 5 different dose rates, being it the control, which is natural background radiation, 30 milligray an hour, 60 milligray an hour, 110 or 430 milligray an hour. Each generation was exposed to a similar, sorry, I'm fighting a cat here. So each generation was exposed to one of these different dose rates, where the parent generation did not have a history of previous exposure. Generation 1 in the previous generation, and generation 2 in the previous 2 generations. This gave us the opportunity to study the effects within one dose rate between the different generations, or within one generation of the different dose rates, theoretically giving us a dose rate effect and a generational effect. Now, after the plants were irradiated, DNA samples were then studied using UHPLC to study the total methylation levels, and a select group of samples, the control 30 and 110 milligray an hour samples, were further studied in depth using whole genome bisulfite sequencing. Our global methylation level results showed that no real changes occurred in the parent generation, but in generation 1 mostly. and we see that the biggest changes occurred at the higher dose rates. In generation 2, we see a shift that's closely resembling what we saw in the parent generation. So it might be a rebalancing. And this has also been seen in other studies, I believe on Daphnia, I'm not sure though, where the first generation has a stronger effect and the following generations it starts tapering off again. The whole genome bisulfite sequencing data showed us that most changes occurred in the CpG methylation context and less so in the other two contexts. This is very interesting, as this could potentially be specific for this stressor. Additionally, CpG methylation has been shown to be very stable and play an important role in epigenetic inheritance. So possibly we might be seeing a heritable effect here. Additionally, we see that most changes occurred in the lower dose rate and less so in the higher dose rates, showing us a potential threshold effect. This has also been seen in other studies in which plants shift from a more acclimating to a more survival kind of strategy. However, yeah, we're not certain yet. Lastly, also here we see that no changes occurred in the parent generation, but in generation 1 and 2, indicating that there is an initial need for an initial exposure to the stressor to have an effect on the following generations, possibly seeing a form of stress priming, which has also been seen as a result of UV radiation and cold stress in plants. When we looked further into the location of these differentially methylated regions, we saw that a significant number was associated with genes, either in the promoter region or the gene bodies. And additionally, we saw that a significant number is associated with the transposable elements. Here we see that hypermethylation is slightly there's a slight bias for hypermethylation over hypomethylation, which has also been seen in other studies, especially in the transposable elements, which was hypothesized to have a genome protective activity. Now, when we looked at the genes that were actually associated, sorry, cat again. We saw that those that had more DMRs in the promoter associated regions showed a higher level associated with processes involved in alternative RNA splicing. Alternative RNA splicing is often used as a dynamic tool to adapt to changing environments and stressors, generally used by plants to overcome these issues. The fact that we see a clear association here could also indicate that this might be a stress response. Additionally, in the genes that had differentially methylated regions in the genome body or gene body, there were processes involved in DNA repair mechanisms or DNA damage response. Additionally, chromosome organization and thus epigenetic regulation. We also saw that a couple of developmental genes were affected, some associated with cell wall formation, which has also been seen to be affected by different stressors, including ionizing radiation, in which the cell wall thickens as a protective method. In addition, we saw that trichome branching was affected. Trichomes are the little hairs that you can see on the leaves and stems of plants, and they have been associated with stress response as they accumulate various antioxidants, such as diterpenoids, and help in various stress response methods. Associated to this, we also saw that plastoquinone and tetraterpenoid biosynthesis was affected. Plastoquinones are associated with the photosynthetic system and help in the protection against photooxidative stress. And the tetraterpenoids contain various antioxidant species, including carotenoids and lycopenes, which have been shown in other species to also be radioprotectant, thereby showing us an antioxidant response to the ionizing radiation. And lastly, we see that there is an effect on flower development, which has also been seen in previous studies, that flower development and floral induction is affected by ionizing radiation stress. And the exact mechanism behind this is still not known, but in my PhD, I'm also working on this. So in conclusion, we saw that no real changes occurred in the parent generation, but in generation 1 and generation 2, indicating a potential stress priming. Additionally, we saw that most changes occurred in the lower dose rate and not in the higher dose rate, indicating a potential threshold effect or possibly a shifted response in time, which we might not have fully picked up. Thirdly, very interestingly, we saw that there was a clear preference for CpG methylation, indicating a potential heritable response in the DNA methylation. And lastly, we can see that ionizing radiation-induced DNA methylation is likely not random, as it is as genes associated with or affected by this methylation were clearly enriched for stress response processes, and also transposable elements were very much affected. Which brings me to the final conclusion that DNA methylation likely plays a role in the response to ionizing radiation and possibly to help in the stress adaptation against this stressor. Then I'd like to thank you for your attention, and I'd like to thank all my colleagues at SUK and my university, University of Hasselt, and look forward to your questions.
Thank you. Thank you, Paul and Sandrine, for your presentations. So, are there any questions?
Yeah, I wanted to ask something to Sandrine. Sandrine, you said that you saw, you had a slide where you said that after a number of generations, you saw in, I think it was C. elegans, that there was an adaptive response at a certain dose rate. First of all, how did you see that it was an adaptive response? And yeah, that would be my first question.
So it's not my work. First,
no, I know.
First, it's the work from Jean-Marc Bonzomme and one of his students, Loïc Kevarek. And they saw an adaptive response, you know, by with experiment with cross-condition exposure. So after exposure during 17 generation, they put the control condition in irradiated condition and the reverse irradiated condition in control condition, and then they run again 4 or 5 generation, and then they observe the parameters, and they observe, they can observe that male frequency was still high after for 50 for the higher dose rate condition, and for the lower dose rate it tends to come back to the control rate.
Okay, yeah. So it was typical for the high, and you couldn't see it in so it was lost in the lower dose rate.
Not fully lost. So they say that it's acclimated, and for the higher dose rate, they conclude that it was adapted because it didn't come back to the controller rate.
Okay, yeah. And the other part of that slide, it was, if I remember correctly, the first generations where you didn't see where the transplant was not done. So before, and there I saw in one of the things, it is the different generations. It were the different blocks that you saw. Is that, or was it after 17 generations?
I can come back on the presentation if you want.
Because I quickly wrote something down, but I saw something go up and then go down again.
Yes, you're right.
Yeah.
I think there is two different behavior depending on the dose rate.
Yeah. So are these the different generations? Because it says 3-day transfer, but every 3 days it's a new
generation.
Yeah.
And so we see that the male rate increase for the 50 for the higher dose rate and stay stable.
Yeah.
And it's quite different for the lower dose rate. It seems to be cyclic, not cyclic, but
at least it goes. Yeah, it goes up and it goes down. I noticed it because another experiment that Paul did, it was on Lemna. He has not put it in his presentation, but because we are still going further into the details. But we exposed the Lemna plants for 6 weeks. It was fall, I think, and we saw also in the growth a similar kind of trend. So we saw that we had a kind of an effect, which was higher the first 2 or 3 generations, and then it went back to normal again.
And up again or not?
I don't think so.
No, it seemed to balance out, which was very similar to the control plants.
Yeah. Okay.
Actually.
But there once was a paper. Oh, whom was it? I once had that where it was really cyclic. Oh, it's a long time ago. I should find it back. It was on roses, I think.
For this experiment, I know that genetics will be assessed, but not yet done.
Okay.
So it will be some molecular data for deciphering underlying mechanism, but not yet analyzed.
Yeah, we were. Yes, go ahead.
I was just going to mention it's not radiation. For other models and other stressors, I have heard of that kind of phenomenon where you see the effect, it skips a generation, but then the following generation, it can happen.
Yeah.
I haven't seen like very conclusive evidence that radiation does that, but I know other models have shown that this kind of cyclic, non-linear kind of response. So I just wanted to mention that.
Yeah. Yeah, it's interesting. But I also what we are going to do on our lemna is an RNA-seq because we first wanted to do a bisulfite sequencing, but that didn't work out for a very stupid reason, but that didn't work out. So we will have more mechanistic information on it, but not on the methylation level. No, this is interesting.
Well, we might still get something on the total methylation.
Yeah, on the
but
total methylation normally.
unfortunately, not that detailed.
Yeah. That's very interesting.
Are there other questions?
I had 3 questions for you, Paul, actually. I don't personally study plants, so what's the generation time for Arabidopsis? So how long was a single generation exposed for? Was it days versus months?
No. So generally, from seed to flowering, Arabidopsis takes about 4.5 to 5 weeks.
No, 6. 4.5. It's from flowering, but to seed.
Yeah, sorry.
Yeah.
From seed to seed is 6 weeks. Yeah, you're right. Yeah. And we exposed them just for 2 weeks after they'd grown for 1 week already. So we let them sprout for 7 days and then irradiated them, because we've seen in previous studies that that's the most vulnerable time period for Arabidopsis to be irradiated. So we expected the biggest effects. So basically what we did is we grew a seed for 7 days, irradiated for 2 weeks. So then they're 3 weeks old. Let them grow till they produce seeds again, use those seeds to repeat that cycle for the next 2 generations.
Okay, that makes sense. So I guess overall then your cumulative dose for a single generation would have been fairly low if you're only irradiating them for a few weeks.
I wouldn't say it would be extremely low, because the dose rates that we exposed them to were quite high.
Yeah.
Even for Arabidopsis. So it's much higher than what you would find in nature.
Yeah.
Yeah. So the highest dose rate that we use, the one around 400 milligram per hour, it's a dose rate where we know that it induces harmful effects. So we went from that downwards to lower dose rates, but still it was quite high. And the samples were taken just after the exposure. So we exposed them during the vegetative cycle, and we only sampled after the exposure.
Okay. So did you, I didn't get to read the paper, did you quantify anything physical about the plants, like the size of their leaf or their roots? Like, did you measure anything like that? Was there any kind of plant level changes that you observed?
We have done that. It's not in this paper. We have done that before, and we have seen differences. As I said, for the highest dose rate that we used, you can see differences on metabolic parameters like the antioxidative response you can see, and you can see the you can start to see differences in growth. So the growth starts to go down. For Lemna, our other model species, we know that actually there is a kind of cell cycle arrest where the plants just stop dividing. And depending on the dose rates, after you put them back into control conditions, they either will start dividing again and pick up with the control, or they will start to really go down and die off. So depending on the dose rate.
That's very interesting.
Yeah.
And then my last question was, I was just curious, have you ever considered or have you done a model where you do this kind of irradiation and then however many generations later you challenge them with a very acute dose to see, you know, are they, if they're more stress susceptible, will they respond differently than one that was never exposed? I was just curious if you've ever looked at that.
we are doing that, but the results are not conclusive at the moment. So we're doing that with seeds that we gathered in Fukushima. So we gathered there Capsella seeds, Capsella bursa pastoris, which is shepherd purse. It's a close relative to the Arabidopsis plant that you see here, but with a heart-shaped seed pod. You will probably seen it because it is distributed all over Europe. But also it's also native to Japan, and so we have had seeds from Japan brought to us, and we are challenging them at the moment. Well, there's a bachelor student finishing up this study at the moment, where we exposed them to different dose rates and looked at both whether or not there's a shift in reproduction, and whether or not we can see whether these ones are better or not better prepared for the additional radiation. Yeah, that's something that we are doing. Yeah.
Okay, that's very interesting. I'm excited to see when you publish that. Thank you.
It will take a while still, I'm afraid. Yeah, I was something that popped up for the slides that you showed now that I never thought about before until I was in this AOP work group on reproduction, is the floral induction. Might that be something, do you think it's due to the fact that the whole plant, or only the apical meristem and therefore the reproductive cells are being exposed or not?
Well, I think personally that it will have an effect that the entire plant is exposed, because currently, like, you know, we're doing studies on the gene expression of certain flowering genes in the different leaves. We do see that certain genes automatically aren't expressed anymore in the older leaves. However, there's plenty of flowering genes that are still expressed in the other leaves, the newer leaves, that will have an effect on the flower induction. Some of them have been associated to be epigenetically regulated, so yeah, that might still be interesting to study, being it either via DNA methylation or microRNA regulation. So I do think the entire plant will have an effect on it, maybe not the older leaves necessarily, but the newer leaves definitely.
I guess this is more of a theoretical question, since I'm not a big plant person, but I was just curious from a more evolutionary perspective. What I imagine the difference of a plant versus an animal is, an animal can move, right? If it detects stress, it will try to move away from the stress, whereas a plant is fixed. So I'm just curious, are there other evolutionary kind of systems that plants have evolved? Will they like send out more pollen, for example, or are there those kind of stress responses that you know in other models that you could maybe look for when you're using radiation?
Well, there's there's been a lot of studies done on flowering and other stressors, not so much ionizing radiation yet. So that's why we're looking at it. But UV radiation and cold and heat, they will all have a certain effect. So in certain conditions, plants will start flowering earlier in order to create a new generation as quick as possible. However, there's also conditions, I don't know them by heart right now, but in which they will wait and will flower much later. Therefore, just sitting there waiting for the conditions to improve and hopefully get the next generation out. The funny thing is with ionizing radiation, the exposure to chronic radiation or an acute dose will also have a different effect. So generally, chronic irradiation leads to earlier flowering. However, acute doses with similar total dose will lead to late flowering. So yeah, we're still looking into that. We're doing chronic irradiation now. Generally,
does that make sense to you? Like, because just intuitively, if you told me that, I would have guessed an acute irradiation, the plant senses trouble and will try to hurry up, right? What you're saying is the opposite. So that's, again, I really am not an expert in this field. So I'm just curious if that makes sense to you, or is it kind of counterintuitive to other stressors?
I think it's maybe due to the fact that the acute radiation is in one go a massive dose. Maybe making the plants think like, okay, the conditions are really bad now, we're not even going to try it. Whereas with chronic irradiation, maybe there's been some, or for certain stressors, seeds are more resistant to stress protection compared to a normal plant. However, with irradiation, of course, that's different because it penetrates everything. So I'm not so sure yet why certain things happen. Maybe it's as a similar response to UV radiation, that it triggers similar pathways.
Yeah, I think also, so the way of escape for a plant, so as it cannot move, it often changes its seed production. But I think if the damage goes too high, like in a real acute exposure, then the plant just goes into shutdown because it doesn't want to multiply or to cell division. Whereas when it's a chronic dose, this might trigger a number of metabolic pathways that hurry up seed production, because it is known for drought stress, for UV stress, like Paul said, and for a number of other stressors, that is exactly what it does. So, and a number of these pathways are DNA methylation induced, and a number of them are also microRNA induced. So that's a number of these pathways are really regulated by fast epigenetic responses like microRNAs. And we have seen, for example, there is one microRNA 169, which is known to be very, which regulates one of the transcriptor factors that goes to flowering, for example, and seed production, and that is induced by many stressors. And we have also seen it being induced under ionizing radiation. So yeah, that is one way that we think that this might be regulated. But yeah, indeed, plants cannot run away, so they try to. The seed normally is quite resistant to stress, so that is their safe haven, because the seed has a seed coat. It is dehydrated normally, and it is therefore not metabolic active. And for most plants, it can stay in that state until there's a new season or a new, hopefully better time. So yeah, the plant doesn't know that radionuclides do not disappear, but drought, for example, disappears. Therefore, yeah, seeds are often the safe haven, as I said, and also seeds can resist quite a lot of radiation. That we have also tested on wheat seeds, actually.
Very interesting.
Yeah, I think it's also interesting to further dig together with Sandrine and Jean-Marc into this into this recovery, because we have done also a lot of experiments where you can see indeed that we can show recovery, like you had what you said with the low dose, that it returns to the control levels. And I'm really curious to see whether we have a high dose where we didn't find the recovery. I know that we've had it in these lemnas, that the high dose did not recover. Yeah, so I think that is really something that crosses the boundaries of plant and invertebrates, which is interesting.
So just out of curiosity, has your group been interested in looking at more higher LET radiation types? Because I think that could be an interesting question too, because if you really want to stress the system out, don't expose it to gamma or photons, right?
Yeah.
Heavy ions would induce a whack of damage. I'm just curious if you've looked at that.
Yeah, we have had, but the problem is with high LET that you have to be able to do it. And so we have not been able to do it so far. But it is, for us, it's interesting within the space research. There's a lot of space research going on at SAK, therefore not so much in our group, but we slowly or surely also having a little bit in that. And there it would be interesting from an environmental point of view. Of course, yeah, we don't have the high LET that much here on Earth.
It is something that you could also study at the microbeam in Cadarache, you know.
Yeah, I know. I have to. I told you the last time, Christelle, that I am really interested. Also, I'm really interested to see whether we can on seedlings that we can grow on small agar plates or something, that we can specifically only irradiate the apex to see if we only irradiate the dividing cells. Whether you see the same kind of responses, because I'm really curious for that.
Yeah, I think it could be possible. We have to discuss more about
yes.
And also, Paul, so I wanted to ask you, because this morning we had this discussion about the fact that epigenetic mechanisms seems to be more important for low dose rates than genetic mechanisms. So do you think that your data confirm this hypothesis? Because you said that you have more changes in the low doses than in the high dose rates.
I'm not comfortable saying that that's definitely true. Yeah, I'd like to do more studies first before I can really say anything about that, but it could definitely be an indication, our findings. And yeah, like Nela said, we're now working on Lemna, which is a faster growing plant, which has more generations, and hopefully we'll be able to see something on the differences between the different dose rates, because there we have very low dose rate, more approaching Chernobyl type of dose rates, compared to 75 milligray an hour, so that's more on the higher side, even for plants already. So hopefully we'll see something different in there, but yeah.
Yeah, I fully agree with you, Paul. So the two dose rates that we tested, so the 30 and the 110, we saw a difference in them, which was surprising. But both of them are still high dose rates. So therefore it's hard to say whether it is really important for whether this difference is an indication that epigenetics plays a more important role in lower dose rates. The only thing that I would say is that as long as I think that it plays a more important role at those rates where the damage is not detrimental. So I think at 110, it starts to be detrimental. And there, I think it's just, like I said before, total shutdown. Whereas at the lower dose rate, where the plant still has a capacity to recover, there you will probably have the epigenetic. Although I must say that even the plants at 400 milligrays, they went to seeding. And we had a quite good yield, not showing much difference except for a little bit lower growth rate. They were still alive. It's not that we killed them off.
Okay. And also,
Just out of curiosity, what is the LD50 ranges for these plant species? Like, how resistant are they?
Arabidopsis is one of the most, that's really our biggest problem, actually. So Arabidopsis is what they call the mouse of plant biology, because it's the most studied plant, because it has a very small genome, it's completely sequenced, you have a lot of mutants that you can just order any mutant and study it, but it's quite radiation resistant. So to put an LD50 on it, I wouldn't even know, because I've never exposed it to a dose rate that showed an LD50. So I never got it killed by the dose rates that we applied. On the other hand, for Lemna, the LD50 was around, I think, 800 milligray. For after 7 days. So, but again, Lemna on 7 days, it has 3 vegetative generations. So we never tested it. We never did the dose rates in one generation. One generation of Lemna is around 2.5 days. But it's vegetative, which I think is something that we either today, but maybe better tomorrow or tomorrow over. Also, I'm really curious about to see whether clonal organisms like Daphnia and Lemna behave differently compared to ones that reproduce through sexual reproduction. So, but for the Lemna, we could really put up a dose response curve.
So you said 800 milligray per hour for Lemna?
And I think it's very hard because it depends also on whether it's chronic, whether it's acute, whether it's during the vegetative state or the floral. One that I really would like to do is to see once that you have your flowers induced, whether the plants are more sensitive or not, a little bit like, yeah, to see at what life stage is the most sensitive. That is something that has not really done before.
Is your group interested at all in looking at radionuclides, like that kind of an exposure route? Because I would assume then it's in the soil and it's taken up by the roots. And there's a biodistribution like a human, like a person.
We have done a lot of experiments in the past, mainly with uranium. But then it stays, of course, in the root, but also with americium and strontium and At this moment, we're doing some lemna studies on cesium and strontium. That's another PhD student that's doing that. Yeah, so, but that's again lemna, where it's not really accumulating in the roots. It all goes. It's sucked in lemna. It's duckweed, so it floats on the water, and the root is actually not really active anymore. So the uptake is actually through the leaves rather than through the root that is just hanging there. So yeah.
Arabidopsis, we've done some uranium studies, like you said.
Yeah.
So,
Yeah, a lot of uranium studies on Arabidopsis, both at different pH values, looking at the interaction with different ions, mainly calcium. We just have a paper out on the role of calcium on the toxicity of uranium. Yeah, it's yeah, there's a lot of work. Not so many people studying plants.
But congratulations for everybody for your presentations. A lot of work on this heavy
Yeah, I really
experimental work.
yeah, I really think I saw some overlap between what you presented, Sandrine, especially on the long term, and maybe tomorrow, Paul, you can show the slide on the growth. It's not published work yet, but I'm sure that we will, even when it's recorded, we can still publish it afterwards. To see, because I think it's very similar to what you have been showing, Sandrine.
Okay. So shall we move on and try to address some of the questions that were distributed with the program? So I'm sorry, but I don't have any internet connection, so I cannot share my screen. If you have the last version of the program, normally you have the question.
Hold on, let me see. I had it open.
Me
It's
too.
because of the security.
I have notes on mine, but I can share the screen if that would help.
I have notes
Oh,
on mine too.
Ah, great.
We're group C,
Group
aren't
C,
we?
yeah.
Yeah.
Group C. So we can, well, we have proposed those questions, but of course there may be other ones that may be interesting. Huh?
Well, we were already answering this question.
Yeah.
Yeah.
Yeah, I think it's the cross-species comparison is very interesting. And also something that we talked about a lot in the AOP working group on reproduction, whether or not, yeah, for me it's still, I still wonder, and also what you asked before, how plants can cope with the fact that they are completely different from animals, and still we see similar kind of results.
I would argue though that what you're doing, like generally, I don't think an animal would be stressed by like physio or whole organism stress response to an increased dose rate, right? It's invisible. They don't sense it at the cellular level. Things are happening, but to the animal, it's fine, right? If you suddenly change temperature, then an animal may move to a different area. So I guess it really depends on the context, but it is a interesting question about how do they cope and what kind of adaptations they have.
Yeah, except if it's radionuclide uptake and it starts to interfere with metabolism or with a certain organ or something like that, then of course, if like if it's uranium and it all goes to the kidneys or something like that, and you start to have kidney problems, but then it's of course very high dose rates. Yeah.
And for example, the work that you presented on transposon, I think it's quite specific to plants or
transposable elements. Yeah, I know that plants of very transposable element rich compared to animals. But it's well known that transposons are methylation induced. For example, we all know these blood oranges that we eat. So with the red fluid, the anthocyanins in them. This is a typical transposable element induced response. So you need to, at a certain moment, you need to change the, I think it's temperature induced, and then the transposable elements start producing these red cells, and then you have red oranges that you can eat. The same with, there is one variety of grapes that I know of that can be red or white grapes, and that's purely based on a shift in transposable elements. So yeah, indeed, the transposable elements are very strongly expressed in or very active in plants genomes.
And also, so you mentioned that the seeds are highly resistant. And can we make a direct comparison with sperm cells, for example, where DNA is highly compacted? And can we sort of generalize the sensitivity of the different stages, development stages, or among?
Oh yeah, but seeds are no sperm cells.
No.
They're actually very small, dried out embryos with a lot of nutritional extra tissue around them. So yeah.
Yeah, I think it's that extra layer around them that extra protects them against most stressors. Irradiation is different, of course, but yeah.
And I think also for the seeds, it's important that when they're dormant, they're dried out. There's no water in them or barely water, and they're metabolically non-active in their dried state. And at that state, they're quite resistant. I don't know about any studies where they have looked at the seed resistance during seed ripening or during the first germination stages. I've never seen a one on that for irradiation.
It sounds like that's conserved because if I'm not mistaken, I believe both tardigrades and yeast can go under anhydrobiosis, and then they are extremely resistant once they reach that stage. So it sounds like there's some parallels there.
Yeah.
Yeah, it's like in Daphnia, you have also this.
But I also know there are some studies on pollen, which is the male part of the plant, and especially if you if you remember your basic biology, so the pollen will enter and then it makes a tube to reach the egg cell or the haploid cell within the part of the plant that will start to make the new plant. And pollen tube is quite a formation, so that is quite sensitive to irradiation. So that is something that we have that we didn't look into it, but that I've read in literature. But yeah, cross species comparisons. I'm really interested in looking at the difference between clonal and non so vegetative or clonal organisms, which I thought showed higher methylation differences over different generations. If I remember what the work that was done at IRSN on the Daphnias as well, which are also clonal organisms, compared to sexual reproducing organisms, where you have, and I think that that might come to the fact that you do not have a real reprogramming in the clonal organisms. So anything that happens in the first clonal generation might be more easily transferred to the other. But that's hypothetical, so that's me thinking wildly.
In fact, the data that
I don't know, Sandrine, what you think about that.
it's in the first generation.
Yes, not. I think it's difficult to compare with such different strategy, because male. I think the male compound in sexual reproduction is a key point that we cannot consider for instance, parthenogenetic reproduction.
Yeah.
It's also in line with question 3, because is there a best suited?
I cannot hear.
Following your
You're very, very low, Christelle. I don't
Yeah,
hear you.
I cannot hear you.
Yeah, okay.
It's
That's
okay.
This is
better.
better.
It's better.
Because on the fact that we can study organisms such as Daphnia with an asexual reproduction, it can be an advantage on one hand, because as it's clonal, it may be it may be easier to detect any genetic damage, because you have a low generally a low rate of DNA variation.
Yeah.
But on the other hand, if you want to study, of course, male versus female sensitivity, it's very difficult.
And do you know whether that kind of studies have already been done, where they looked at the mutation rate over the different generations in comparison with the methylation changes?
So
In Daphnia, I think Marie Trigeot did that, no?
it was not the same, because there was a work that you presented from Florian Parisot,
Yeah.
where the 3 generations were exposed continuously to different dose rates of gamma external radiation. And in fact, so the parents were exposed from the egg stage until the adult stage. And in F1, there were less damage than in F2 and F3. And it was interpreted as a higher DNA repair in the F1 generation, which was followed by a lot of accumulation of DNA damage to the following generations. And versus the work of Marie Trigeot, who studied DNA methylation in Daphnia, she just irradiated the parents. And then she followed DNA methylation over the successive generations. So there are in fact, it's always what we discussed also this morning. So there is no study where genetic and epigenetic effects have been analyzed at the same time.
Yeah.
Because it's hard work to do that using,
It is hard work, but I think it's very interesting.
And very expensive.
Yes.
Yes, that too.
My argument, though, if you had all the money in the world, would be how do you answer that question? Because if you go very high dose, you're going to definitely impact genetics. I feel like at that high dose, you're going to cause so many adverse reactions. It's going to be a mess epigenetically. Versus low dose rates. Low doses are good for epigenetics, but then they're so low that you're not inducing those genetic changes. I don't know if there's a good model system, maybe like a specific species that would be ideal to study, but to me, that's always going to be a challenge, right? Where do you pick the dose and dose rate to get a balance?
So what I would do and what we're sort of trying to do with the Lemna is pick different dose rates, picking one high dose rate, one very low dose rate, and in between we select a couple to see if you might perchance find that switch moment, because previous studies done in our lab have sort of made a dose response curve of Lemna, where you saw that in the beginning or the lower dose rates, you had a more acclimating kind of response, and then at a certain point, it just dipped and it was pure survival. So I'm guessing that if you go to studying the genetic and epigenetic at the same time, you might also have to see that switch moment happening. The thing is, noticing a switch moment in genetic and epigenetic changes might not be as clear cut as seeing antioxidant response, probably.
I just think realistically, you're going to spend so much money trying to look for that switch, right? That
yeah, but you gave me unlimited money. You said
okay, fair. I set myself up for that. Okay, that's fair. I think realistically, though, that's where it became a problem, right? You can't run a dose response with 20 doses of dose rates, right? You have to pick and choose a couple for feasibility's sake.
Well, what we have now, we're quite lucky in having such a facility, is that we have a panoramic source where you just move away from the source, and then you have different dose rates going on in one go. The thing is, you need to have quite a lot of biological material as well to keep, or to have sufficient sample as well. But this is something we regularly have troubles with, not having sufficient material to study everything that we would like to.
Maybe interesting for the question 2 on cross-species comparison is maybe I can show you a few slides tomorrow on that. So what we have been doing is looking at recovery capacity of Lemna and comparing it to planaria. So planaria are these organisms that you can cut off the head, and you look at the it's starting to rain here. You can look at the tail that forms again, or the head that forms again in the other one. So yes, I do have slides on that. And I can try to see. It's a year ago since I looked at these, so I have to look at it again to see how much comparison we already did. But it was a master student that worked on it, and it's a topic that I now still am following up. But I can show you a little bit on the comparison between the planaria and the lemna that are both regenerative and fast growing. This is, that might be interesting. I have to go in a few minutes. I don't know, Christelle, how you see us progress, or shall we go through the other questions tomorrow?
Yes, we have the whole day tomorrow to go on. And just perhaps another remark regarding asexual versus sexual reproduction. It's something that Fred Alonso tried to do in Daphnia, because you can shift to sexual reproduction in Daphnia when the conditions are harsh. And it's true that if you change the model, you cannot compare. So the best way is to do that within the same species. So I know that he tried to do that, and it's something that could perhaps be redone, because it did not succeed in having sufficient males. But also perhaps some data can be reanalyzed, because also in nematode data you can have a shift between
Mhm.
hermaphrodites and males. And in fact, we have several data. Perhaps they could be analyzed further.
And sorry, Christelle, if I'm not mistaken, C. elegans can also do that, right? They are typically, I believe, hermaphroditic, and then under
Yes.
very stressful conditions, they will undergo sexual reproduction, or maybe it's the other way around. But
Yes, yeah.
has your group looked at that? If with chronic irradiation in C. elegans, if they shift their mode of reproduction?
In fact, it's what Sandrine presented on the male frequency.
In
Oh,
fact,
right.
in fact, by irradiation, with irradiation, male are generated from the different strains. At the beginning, I think the level of male was 10 or 15%, and it reached nearly 40% after irradiation, multiple generation irradiation.
Yeah, something that I learned from the work from your PhD student, Sandrine, is that in strong contrast to us humans in C. elegans, it's the amount of male cells that is limited, whereas in humans it's oocytes that are limited. So you're born with a number of male cells as a C. elegans, and you're born with a number of oocytes as a female human. Whereas, yeah, that's true, isn't it?
Yeah, it's true, but they have the capacity to increase the number of males when they are in danger to compensate this limited number of sperm.
Yeah.
So it's not.
It's not the same as oocytes for humans.
Yeah, it's not the same. First, it's not the same, and they are they are able to adapt themselves, and it's not only with irradiation. It's known also for thermal stress or other stress. C. elegans is able to increase the number of male when there is a high stress in the environment. So yes. And for crustacean, for us, we also run an experiment on Gammarus forsarum, because we wanted to try to compare with Daphnia. In fact, that is a parthenogenetic organism. And for the moment, we don't yet have analyze fully the result, but male are more sensitive than female in this crustacean species.
Yeah. Interesting.
Very interesting, yeah.
Yeah.
So
okay.
I don't want to
Christelle, how do you?
But I think it's the difficulty for cross-species comparison, because in the environment, the strategies of reproduction are so different that it's difficult to compare fully. But we saw with the AOP group also that finally the different step of adverse leading to adverse outcome are not the same fully, but the steps are very similar. So it's quite interesting.
Yeah, that I thought was quite striking on what the AOP group did. Yeah.
And I think that Cisco will talk about that tomorrow on the different reproduction strategies among the biota. So
that's interesting.
Yeah.
And before I forget, because I had a remark when Sandrine, you presented the work. I was thinking about the dose calculation over transgenerational or multi-generational studies, because it's something that we always say, okay, Daphnia have been exposed for 1, 2, 3 generations, and we see a decreasing EDR10, or I don't know how to say, but the effects appear at lower dose rates through
Yeah.
the generations. But in fact, how should we calculate the dose? Because in F2, they have been exposed for 2 generations. You know, there is a sort of historical accumulation of dose. So is it really good to use the current dose rates and not the dose rate accumulated by parents and grandparents and
I totally agree with that. Even more with the result we had on frogs in Chernobyl, because I'm always wondering if we are not missing a part of the issue when we just look at the ambient dose rate or reconstructed dose at the present time. For me, it's the history of the dosimetry needs to be considered, and I don't have fully the solution how to do that, but
No. Yeah, it was something that once when we wrote together this adapt project for Euratom, it was how's his name, Germán Orizola from Spain, that suggested to study the Przewalski horses in Chernobyl, because they were not there when the real high dose rate was there. So they were only exposed to the current low dose rates because they were only released, say, 5 or 6 years after the accident.
Okay.
Yes, there are some papers that have been published on that topic, recalculating the historical dose and
the one of Karine, no?
Yeah, among them, but also for rodents and Drosophila.
But I think it's a good point to submit for me.
Because it's something that is completely different from humans. Completely not in fact, because among the workers, but generally they can escape the radiation, and there is no cumulative exposure.
Yeah, although in some of the presentations this morning, also the transgenerational in humans, for example, the bomb survivors, they had a very high dose in a previous generation. Okay. I'm sorry, but I have to leave. As I told you, a former colleague of our group is retiring today, so I still need to go to a reception.
Yes, have fun to the reception. So we talk a little bit about definition. What is a multigenerational exposure? What is a multigenerational effects and stuff like this tomorrow? Make it precise, or we fully agree what is behind these words?
That's good. That's good.
Is there anything else we should be working on ahead of tomorrow? Like anything in particular we should prepare? Or
we didn't look into that.
Yeah, we have the questions to guide us. I'm just curious if there's anything in particular.
Yeah, I've just taken some notes for all the questions, at least to what I could answer. But yeah, we'll see tomorrow.
Okay, that sounds good.
Yes, there are, I don't know how many, but I think there are 14 or something like that questions.
Yeah.
13 questions. So we have just addressed, I don't know, one or two.
One.
So I think tomorrow we will have the time to go deeper into these questions. And
Yeah, I think we can go for tomorrow. We can go one by one, and if you have something interesting, also you, Cheyenne, if you have anything that you think, this is very interesting for a certain question, just prepare a slide or two, and we can pop it up and discuss it. I think that's the nicest way to do it.
Okay, that sounds good.
And perhaps Cheyenne, as you send me your files, some files on the systematic review that we are planning for the TG for this task group, perhaps that we could discuss on that also, on the search terms, on for example, we talked also about adverse outcomes. What are the important adverse outcomes to consider, things like that, and this could perhaps enrich also our thoughts for the systematic review.
Yeah, that's a good point. I'll have some slides prepared for discussion for tomorrow.
Okay, perfect.
That's fast.
Thank you very much for joining.